So now that we've looked at the history of natural selection, let's look at the exact mechanisms in which natural selection can work to change allele frequencies in populations. First off, think of natural selection as filtering out certain alleles. So there has to be some sort of filter that allows some alleles through and other alleles get left behind. Using this image here of a filter as an example, in natural selection, it's the environment that acts as a filter. So only the fittest individuals can survive and reproduce in a given environment. Think about it this way. If you have organisms that are adapted to live in an aquatic environment, such as a lake or a river, if you take them out and put them on land, will they survive? No, because they need to be constantly wet. So they are not adapted to live currently in a terrestrial environment. So think about natural selection simply as the environment acting as a filter in order to change allele frequencies within a population. So we can think about this in a very simple way. Looking at these axes here, on the y-axis we see frequency of individuals within a population, and on the x-axis is a range of phenotypes. Remember what a phenotype is. A phenotype is a physical manifestation of a genotype. So what an organism looks like, what its behavior is, etc. But a phenotype has a source in the DNA. Okay, so let's take this as our standard bell curve. So in a normal situation, we have most individuals of the population falling somewhere in the middle. So there's some intermediate phenotype where most individuals of a population lie. On the extremes, or on each end of this bell curve here, we have less of the population. So you generally have lower numbers on each of these extremes of some phenotype and more somewhere in the middle. So this is the baseline. So natural selection can adjust this baseline based on a given environment and the variation already present in the genome of organisms. There are 3 different ways that natural selection can work. All of these are relative to the bell curve we just drew. So think about how each of these are working relative to this figure here, a standard bell curve, where most of the individuals are in the middle, and you have fewer at the extreme. So the first type is called directional selection. This is simply where you take the bell curve and shift it in one direction or another. The classic example of this happened in the industrial part of England in the 1800s, where there was a lot of factories burning a lot of coal. And as a result, trees became covered in soot. Prior to that, moths here generally came in a range of colors varying from white to some sort of mottled gray to completely black. Those are our phenotypes. And most of the population tended to be peppered, a sort of grayish white color. And that meant that the white ones stood out on a tree and would get eaten by birds. Black ones stood out on a tree and would get eaten by birds. Therefore, most of the original population was peppered. Once the Industrial Revolution came, all the trees became dark because they were covered in soot. So if you think about our 3 original colors, white, peppered, and black, which ones are going to survive? In this case, the black ones would survive because the black ones could blend into the trees. The peppered ones and the white ones were very obvious and stood out So any passing bird would eat them. So in other words, the genes that coded for the phenotype of black or peppered or white were adjusted by individuals being picked off by these predatory birds. As individuals died because they were removed from the population, only the alleles of the survivors remained. This is what survival of the fittest means. Those that were able to live and reproduce in a given environment are fit. In the case of the original group, the peppered moths were the most fit. Once the industry came and polluted the trees, the black ones became most fit. But as the Industrial Revolution wrapped up and a lot of air quality measures were passed, the air quality got better, and trees that were around weren't black anymore. So the curve shifted back, away from black ones being the most fit. In this case, once the trees became lighter, the peppered ones and the white moths became more successful, so they became more fit. Versus black ones, which were very obvious against the whiter tree and got eaten by the birds. So directional selection takes our original bell curve and moves it in one extreme or another. Stabilizing selection, on the other hand, takes our bell curve and the environment selects for the intermediate phenotype. So the extremes disappear, but the intermediate phenotype remains. There are very few individuals at each extreme relative to even more being at the intermediate. So in this example here, we're looking at a particular type of cactus. So the number of cacti, so the frequency of cacti here on the Y, and the number of spines on the X. And as you know, cacti have spines. If a cactus has too few spines, less than 80, for example, pigs, these peccaries here, would eat them. So you're removing individuals from the population that had very few spines. On the other extreme, too many spines allowed a home for a parasitic insect. So in this case, too few spines meant if you were a cactus, you were likely going to be eaten by a pig. Too many spines meant you were going to be parasitized and killed. So individuals with a lot of spines or too few were removed from the population. Therefore, the gene pool shifted to favor those that had an intermediate amount of spines that wouldn't be attacked by a pig and couldn't be parasitized. So in this case, our bell curve shifted to be skinnier and taller, with very, very few individuals left in the extreme. Another example of stabilizing selection is what we call heterozygote advantage. And we've talked about this in prior units where we talked about sickle cell anemia. If we look at individuals with sickle cell anemia, which is of course a disease of the red blood cells, sickle cell anemia seems to overlap in areas where malaria is common. And this is because stabilizing selection is at work. Individuals who have sickle cell anemia, meaning they have all of the genes that code for complete sickle cell anemia, meaning 2 recessive alleles, means that you are going to die from sickle cell anemia. If you're an individual that has 2 dominant alleles, meaning normal blood, you're going to die of malaria. Individuals that have the heterozygote, 1 dominant and 1 recessive, have essentially partial sickle cell anemia. You have some regular blood cells and some sickle cells. So you don't die from malaria, but you also don't die from sickle cell anemia. So again, stabilizing selection is a favoring of the intermediate phenotype in an environment over the extremes. So by logic, that means our final type of selection is called disruptive, where the intermediate phenotype disappears or has a less frequent number of individuals relative to the extremes. And this has happened on the Galapagos with a different species of Darwin finches. And again, it overlaps very much with environmental conditions changing. Intermediate types of beaks were not favored in this case, as you only had plants giving large seeds or small seeds, but no intermediate amount of seeds. So birds that had intermediately sized beaks did not survive in the environment. Only those that could eat very small seeds or very large seeds survived. So you had a break in our bell curve. Each extreme was favored. And this is a nice summary slide right here to sort of talk about those three different examples. Here we have our original group. For example, we're looking at mice here. So imagine mice in different environments that have different types of fur color. You have a standard bell curve. In this case, this one on the far left, you have stabilizing selection, where you have this sort of tannish mouse that is able to survive. It could be on something like sand, maybe. But the extremes disappear or become less frequent. Directional selection is where you move the curve to each extreme. Imagine it could be a type of rock that's very dark. So you are removing individuals from the population that don't have the dark color fur. And the ones that survive are darker colored, and they pass on their genes. Versus disruptive selection, this last one where the intermediate phenotype disappears, and only those on the extreme can survive. I briefly talked about this before with the last video, but what does survival of the fittest really mean? It means that an individual can survive in a given environment and reproduce. Fitness is survival and reproduction. Fitness does not mean strongest. It means ability to survive and reproduce in the environment. And this is where the term adaptation comes from. Adaptation has to do with surviving in a given environment. Sometimes that means running faster. Sometimes that means having a longer neck in the case of a giraffe to reach more vegetation. Adaptations occur due to natural selection for a given environment. But natural selection is not meant to fashion perfect organisms, and it is completely misunderstood that way. Natural selection is particular to an environment, and it can only work on what's existent in the genes, what's already there. Adaptations are simply compromises in many cases. We can look at some of these deep sea fishes, which are very strange looking. But it's an accumulation of different types of adaptations to live under high pressure environments, to live under low light environments. The idea of evolution is not to create perfect organisms. That is not what evolution is. Evolution is the ability to survive and reproduce in an environment. And clearly, the fourth point here is the key. Chance, natural selection, and the environment interact to create adaptations to allow individuals to be fit.